So welcome back to the 8th meeting of our class, Engineering 443-543. And I need to remind you that, as usual, this is being live streamed and recorded. Although I do edit out the preambles and the chat before and afterwards. But during the lecture proper, it's recorded. And today we're going to continue our discussion of diffusion and decay and diffusion limited growth. And we'll see how far we get. There were some, only 2 people had it in the homework. And so I looked at the homeworks, and there were a few things that might be worth going over in a little bit more detail. So people should feel free to say that we should spend some time doing homework problems if there are questions about it. And there was one thing that I noticed that I think is worth mentioning that I'll come back to in a few minutes, which is something that I normally lecture on in the fall, but may be worth mentioning again today.
So I can at least speak to that for the homework. Last lecture clarified a lot of things for me, so I redid the problems, and I made it through pretty much everything, except that for the last problem, I don't know why my simulation keeps crashing, and I've remade it from scratch twice. So that is why that's where I'm stuck, at least.
Okay, we can walk through that together. I suspect that Twinit laid a little bit of a trap for people, which is that the default boilerplate code for cell division that's pasted in when you turn on in Wizard assumes that you have 2 cell types. And if you create a simulation with only 1 cell type, it then tries to change a cell from the type that you have to a non-existent type, and that causes a crash. And so my guess is that that's what's happening. That error should be trapped. In other words, it should throw an error but not crash CompuCell. And so that's now on the punch list for fixes for the next release. But because what I think was it either Josh or Jackson, I think, asked that question. He said, "Why is this code crashing?" I said, "Oh, you're doing something that was not asked for in the homework assignment, but is boilerplated in, so you have to take it out." So that's a good one. But we could come back to that a little later.
I'll also try to go ahead and alter that really quick to see if that fixes it.
But my guess is that there's in the cell division plugin, mitosis steppable, there'll be something that says if cell type equals 1, make cell type 2. And if you don't have a cell type 2, that causes a crash. Any other questions before we get started on the lecture proper?
Yeah, I actually also had a question on problem 5. You ask us to make a scalar tracking field for the amount of glucose that gets uptaken.
Mm-hmm.
And I believe I did it right, but my the scalar fields in CompuCell, all I see is the outline of the cell with a completely blue background, which is essentially saying that there's no glucose being uptaken. So yeah, I don't know if that's the expected behavior or if I did something wrong.
So there are 2 possibilities there. We'll come back. We'll do that actually as an exercise later in the class if we get to it.
Okay.
There are 2 things to be aware of. One of them, which we talked about a little bit, is that when you're doing fields, you have to adjust the display to determine whether you have auto-scaling of the colors or whether you have fixed colors.
Mm-hmm.
And the typical default scaling is 0 to 10. And so if the values that you have are very different from that, then you may not see anything because you may be outside of the range of the display. So then you need to auto scale. The bottom of the display, if you look here on the screen, it's pretty small, but I have a chemical field, and it'll give you min value and max value at the bottom.
Yeah.
So you could then look and see whether your color bars make sense given the minimum and maximum values in your fields.
Okay.
Another thing that we've run into before, this was something that people had issue with in homework 5, was that sometimes the sign that is returned by CompuCell for a measurement may or may not be the one you expect. And so when you do uptake total count, it will tend to return a negative value because it's thinking about the amount that's being taken out of the field. But that's from the point of view of the field. From the point of view of the cell, it's a positive value. And so you need perhaps to make the change the sign, to flip the sign of that, so that you get the display that you're expecting.
Oh, yeah, no, that's it. I'm looking at my figures right now, and my color bar goes from 0 to 1, but it says my min is negative like 0.00025, and my max is 0. So, yeah, I think I just need to change this color bar scaling or the sign.
Yeah. But that happens with the pressure too. And so the little pep talk I was going to give you, and I didn't create a slide for it because it really only became clear to me when I was grading the homeworks today, is that I gave you an example, which was to do contact inhibition, pressure inhibited growth. And I gave you some functional forms. I gave you a 1 minus the pressure. I gave you 1 over a constant plus the pressure. I gave you 1 over n over n to the fourth plus the pressure to the fourth. And something you really need to start thinking about, and again, in the fall I give a lecture on this, and normally a lot of people in the spring have taken the fall course, so they've seen it before. But since most people here are new, I should probably give the same lecture again. Is what I would call limiting cases, or if you like, sanity checks. And so if you ask a question like, I'm going to have a rule that the rate of growth of my cell is some function of the pressure, you should ask yourself if the pressure is, first, what side is it? If the cell is being compressed, then it should stop growing. If the cell is being stretched, it's certainly not going to stop growing. It might not grow faster, but it shouldn't be stopped growing. So you have to ask the question, if the cell is being compressed, what does the pressure measurement that I get from CompuCell return? Is that a positive or a negative number? And then I should ask the question, whichever it is, if the cell is not compressed at all, how fast should it grow? What happens if the pressure changes when it's not compressed? Does that make a difference to the rate of growth? The answer is probably no. It might be. And so that tells me that in that region, if the pressure is pulling on the cell, I want to have a function that doesn't depend on the pressure. If the cell's being squeezed, I want the growth rate to go down. And so in that situation, I could have 1 minus the pressure, the force of compression. That would be reasonable. Although in that case, the growth rate will go negative at some point. Should the growth rate ever go negative? Should the cells shrink if they're pressed too much? And that's why those other functional forms are there. But you should look at whenever you have a function, you should ask the question, when I change the argument of that function, does the behavior qualitatively make sense? If I'm quantifying the growth rate of the cell and the pressure and pulling on the cell, then the growth rate shouldn't depend on the pressure. If the pressure is 0, then I have some base growth rate. If I squeeze more, the growth rate should slow down, not speed up. And so if I look at, some people wrote a functional form which was 1 over 1 minus the absolute value of the pressure. In that case, when the cell's being squeezed more, the growth rate goes faster, or it goes negative. And the Michaelis function is 1 over 1 plus the absolute value of the pressure. And so if you ask the question, if I press this on the cell a lot, should it be growing or not? And your function says that it grows faster when you're pressing on it a lot, you know there's a mistake. You may not know exactly what functional form you should use, but you don't have to do much math to do it. You ask the question, if the value of the argument is 0, what happens? Is that reasonable? If the value of the argument is positive and small, what happens? Is that reasonable? If the value of the argument is very large, what happens? Is that reasonable? And if you know those three limits, 0, small, intermediate value, and very large value, if it's well behaved for those three cases, it's likely, it's not guaranteed that it'll be right everywhere, but it's likely to be right everywhere. If it's wrong in those cases, you know you've made a mistake. Now, of course, the space of mistakes is very large, so eliminating one mistake doesn't eliminate all mistakes. But it's very helpful to have some sanity checks. And I think it's important, and sometimes these questions are worded in a particular way, so that you have to ask those questions. Because as the course goes on, I understand we're not all used to asking those questions all the time. As the course goes on, I think it's important for you to try and develop that kind of intuition. Ask the question, does this function make sense? Does what's happening make sense? In the case of pressure-induced growth, or what we call contact inhibition, you can ask a reasonable question. If your cell's not being pushed, it should grow. If the cell's being pushed harder, it should grow slower. If the cell's being pushed a lot, growth should stop. Whether the cell should shrink is another question. That's something you could ask. That's a debate. But one thing you know is that the growth rate shouldn't go to infinity when you're squeezing on the cell. So if you have something like 1 over 1 minus the absolute value of the pressure, if the pressure is plus or minus 1, the growth rate's infinite. And that clearly isn't reasonable. And so those are the kinds of things that I think people can do, regardless of your mathematical background. Some people have a lot of math, some less. But asking a question about, well, is this reasonable, is something that we can all do. And when I build models, the first thing I do before I do any coding is ask the question, what do I expect as reasonable behaviors in these kinds of simple cases? I may not know the exact shape of the curve, but I know that it's 0 at 0, that it doesn't blow up when I go to very large values. Or maybe it's 0 at 0 and it does blow up at very large values. Maybe it goes negative. But those kind of things, those kind of what I'd call semi-quantitative things, are usually things that you have some intuition for. And so don't abandon your intuition when you're using math or computers. I think that that's really, really important. And it's important in the experimental lab too. You do some measurement with some machine gives you a number back. It's important to know whether that number makes any sense. You may not know whether it's right to 10 decimal places, but you should know whether the machine's operating properly or not. At least if it's giving you a number that makes no sense. And so I think that kind of intuition takes time to build, but these kinds of questions are useful. And so looking at things like ranges, scales, sensitivities. So there are a fair number of problems that I've assigned where I ask you, say, try this parameter value and this parameter value and see how they're different. My hope is that as time goes on, since it doesn't take very long to change the parameter value and run it again, that people will more or less automatically try some other parameter values and see how things change. Because the idea is to try to build some intuition about how these systems work. They're complicated. Sometimes our intuition is wrong. But in general, if you break it down into pieces, you can understand the pieces pretty well that way. So, and things like remembering to check what the display range is, that if you're not used to that, of course, you don't think about it. But once you've made those mistakes a few times, and believe me, I've made those mistakes plenty of times myself, it's sort of you build yourself a sort of a checklist that says, gee, did I set the ranges right? Gee, should this be positive or negative? Should it be big or small? Should this thing really blow up? Those kinds of questions. I could give you those checklists, but if I give you the checklist and you haven't done it yourself, you're going to say, why is he making me tick all of these boxes? What's the point? And so I think it's important to try and see what fails, and then use that to learn what makes sense to you, because my checklist might not be yours. Okay. So that was my comment that I wanted to make about the homework. And I'm happy to go over things with people in more detail later. So I think we still are owed a small presentation by Joel and Jackson. And so I hope we'll get that. And then I would like to review a few of the things that we did last time. And again, if people have questions about homework problems, we can dig into those a little bit more. I'm going to do a little bit of algebra because some people had some issues with some of the algebra I was asking people to do. And then I'd like to talk about sources and sinks of chemicals, which is that challenge problem in the homework that was just asked about. And then possibly, although I suspect we won't, I'd like to start talking about chemotaxis. So if we get to chemotaxis today, that's great. Otherwise, we'll do it next week. So why don't we start out, if people have project updates. And Joel, are you and Jackson ready to present a little bit?
Yep.
Great.
Okay. Do you want to take it away and start sharing from your screen, Jackson, and then we can switch?
Yeah, sure. Let me just pull it up really fast.
Cool.
Everyone see it?
Yep.
Great. So we're doing a cell death model. And so these are the types of cell deaths that we were originally going off of based off of the paper in the bottom right corner. The ones that we're focusing on are pyroptosis, necroptosis, apoptosis, and necrosis. Pyroptosis is an inflammatory type of cell death response, and necroptosis is kind of a fail-safe, or if apoptosis fails, then apoptosis is just the normal regulated cell death that generally occurs at the end of a cell's life, and then necrosis is generally just cell death that occurs from like trauma or like toxins introduced to the cells. And then some of the other cells, and then we picked these cell deaths because they have, a lot of them have more of the research on them, even though they're newer. And so it just seems simpler to start with these four, and then if we had time, go on to other types of cell death. This is just to show, this is one of the papers we found that we're going to try to use. We're having trouble finding the code currently, but that's, this is a PetriNet model that they made for necroptosis and apoptosis. And this is just kind of to show how complex the pathways can get and how far off they can change. And then this is to show that they were able to actually show that there are certain molecules that are only meant for those parts of the pathway, and we're going to use those molecules as our key molecules in our model.
Yeah, so we started by using pyroptosis, this paper, a single cell mathematical model for COVID-induced pyroptosis. And the model was very well noted, as you can see by when I move my mouse, it doesn't show anything, sorry. The habit. On this image, you can see the basis of the model. And what I try to show here is that this reaction that we are seeing on NLRP3 is represented on a math models with those functions that you guys are seeing on the right. And from there, we can write code that is doing the exact same thing. And on the bottom, you see a specific type of coding, which is called antimony, that works better in representing in the sense in the community of biomodels. This is the type of language that we use, and it's easy even for us to understand. For example, on the J231, we can see nothing and then an arrow, and then it goes to NLRP3I. It's basically saying from nowhere, it's coming something that is creating NLRP3I. And here's the function that tells which rate is this molecule being created. The same goes from the bottom. The second one is a decay function, and actually we're seeing the NLRP3 going to nothing, meaning it's decaying, and the rate is delta 1, right, times the number of the molecule. And you can see that code on top. If you look at the first equation there, the delta is right at the end, and you guys can see a minus sign. And since the minus sign is not needed, since we are already establishing that this molecule is going to nothing at this rate, so it's doing the same thing. From there, you can go to the next slide, please, Jackson. From there, we start doing models or a representation on CompuCell that I'm about to show you of how it works. The numbers are not correct. It's not calibrated yet. These inside functions are not tuned yet, but we can see some of the behaviors. Can you click it again? Because I just want to, yeah, there's an animation there. This is representing the DAMPs and PAMPs. And the other one is representing the liberation of IL-1 beta. So let me get the share. Let me show you guys in action. So here is the code. You guys are seeing basically the same thing. This is the cell field. All cells here are healthy cells. Here we have a damp spot. And we were going to see some action here, but it's following just the IL-1 beta molecule. And here there is like a cell count. We're going to see all cells starting healthy, and then the dead cells will surpass the healthy cells as the simulation goes on. So here goes. I think it's a good point to stop here in the middle. As you guys can see, the number of DAMPs are increasing, and some of the cells are getting initiating pyroptosis by DAMPs and PAMPs, which is represented by this green cell. When they die, the way I coded it is supposed to release more DAMPs in the system after they die by this type of death. And on the red, we can see cells going pyroptosing, but based on the IL-1 beta. So they are releasing here, and they're telling the cells near it, the neighbor cells, to start pyroptosing, but for different reasons. They recognize the IL-1 beta as the signal to start pyroptosing. So let me keep playing. And you guys will start seeing a behavior where the pyroptose induced by the IL-1 beta seems to take over way faster than the other one. So it represents very well what a cytokine storm would do in the pursuit of limiting the amount of virus or whatever is the pathogen in the body. And we can see the difference between the two here. I previously showed this model to Glazier before. I just changed the point where the cells are secreting. Instead, before I was secreting like 3 points away from the cell. Now I'm just secreting like 2 points, and this already gives a huge difference on how much it spreads in the system. But yeah, that's I'll play again so you guys can see, and also see the cell count. Yeah, so the cell count here is a good measure for us to see how many cells actually start pyroptosis because of DAMPs, and which ones started pyroptosing because of the IL-1 beta. Let me go back to the slides. Can you pull up the slides again, please, Jackson?
Can y'all see that?
Yep. Yeah.
All right. And
So
then, or are you done with this slide? Sorry.
yeah, yeah.
All right. And then, so we're tweaking that simulation. Then next, we're going to try to implement a necroptosis model. And we found in this paper about a incoherent feed-forward loop about the necroptosis decision making within a cell. They were able to create a mathematical model of it. This code here is basically like conceptualizing the initial cell and leading it towards necroptosis. And then this module right here decides whether the cell is going to undergo a necroptosis or not. And so we're going to hopefully, that's the next step for what we're trying to implement. And then after that, we found another model for apoptosis, and then we're going to try to implement that next.
Yep. So basically, those are the next steps that we are doing. We're trying to create. Now that the question is if we create different modules and then add them up, or if create them inside the already existing one, and calibrating is always a pain. And that's the next part that I think will take most of the time. Creating the code seems to be fairly straightforward if we know the behavior that we are expecting. But yeah.
That's great. It's always tricky because the way people think about the problem may be different, and some of the molecular species may be shared between different pathways, but and some of them may not be. Some of the components in this in your pathway may be molecular species. Others could be behaviors of the cell.
Yeah,
Go ahead.
Sorry. One of the things that me and Jackson were talking about is that types of cells also have different characteristics. So if we want to simulate this in a broader range, we should be able to establish or make people choose what type of cells that they want to simulate instead of one single cell.
Yeah, we found
Right.
that
Well,
one
that
paper.
tends to be an issue, that people make these measurements in typically in vitro, and when you've got cells in culture, they're not behaving the way they would in your body anyway. And then, as you say, for a particular cell type, which may or may not be the cell type you care about. And so the cells in your body differ enormously in their susceptibility, say, to apoptosis, their threshold. On the other hand, you have to start somewhere. And the fact that you've got 3 executable models of these 3 pathways is already a lot. So I think it's quite exciting that you've got this.
Yeah, we have to discuss later with you on 101 how we're going to do the necroptosis, because that seems to be the hardest part to find a mathematical modeling of necroptosis, since it's a failure, right? It's hard to describe it.
Well, you may not be able to do everything, but I mean, this is a course. So if you could get a good model working of one of these, that would already be a lot for a semester project.
Stop the share then.
Okay, well, thank you. That's great to see. Does anybody else want to talk about projects briefly? You've got certainly a chance to report on where, if there are questions, comments, updates. Does anybody else have anything that they want to review quickly? Connor and Andrew, I guess your main thing was that you weren't able to talk to Roland, but how is the chemotaxis module working out?
It's going pretty good. We've made a fair amount of progress. Basically, right now we've gotten it so that we can take the patchy gradient and we can move it around within the simulation. And what we're working on doing now is kind of taking the measurement of velocity that we were doing with it bouncing around, but instead of switching the sign of lambda, each time it makes it within a certain distance of the focal point, it moves the gradient. So then we can measure velocity as it goes up that way. The one difficulty right now is that we need to kind of calibrate the parameters a little bit better, because the way it is right now is that when the cell's at the beginning of the gradient, it's having difficulty chemotaxing up it. And then when it gets further up the gradient where there's less patches and the concentration is stronger, it's tearing the cell apart. So that's where we're at right now, working on that. And we also are working on scheduling a meeting with Juliana to talk about persistent migration.
Great.
Andrew, do you have anything you want to throw onto that?
Yeah, just what I've observed is that in the beginning, when the cell is initialized, it starts off with a fast velocity, but then once it gets closer to the gradient, it decreases in velocity like a lot, and so it gets much more difficult for it to actually reach the center of that gradient, that focal point. And I'm not sure exactly what that causes or what's causing that to happen.
Wait,
I'd be happy to meet with you one-on-one about that. That would be fine. We haven't. You're suffering a little bit because we haven't done chemotaxis yet. Maybe this maybe a little bit today, maybe otherwise next week. But I think one of the things that we often do, which CompuCell doesn't have built in and probably should, is adaptation. And so if you divide the lambda chemo by the mean concentration, then you can turn the lambda chemo up a lot. Basically say that as the concentration goes up, the lambda chemo goes down, which is what happens in real cells. And that allows the cells to be much more responsive to a larger dynamic range of signals. But that's certainly, there are other possibilities as well, but that's one thing you can try. Now, it may or may not be true of this particular cell type you're using. But of course, if it's an abstract cell type, then you have the choice. But that tends to make the cells more responsive. And for the other for the other things, I guess we have to see it. I mean, if you want to show it, we can look at it together now, or if you want to meet separately, we can do that.
We were seeing them getting slow, but on the other extreme, they get so fast they disintegrate.
Right.
So 2 reasons to rescale the lambda by the concentration.
Yeah, I think that'll probably solve it, because that was the conundrum of trying to find a Goldilocks zone for a static parameter at that point. So yeah, I think after we do that and meet with Giuliano, we'll be at the point where we're actually ready to start putting multiple cells and then trying to get the ligand binding site set up.
Great. Okay, that sounds good. Wonderful. Josh, did you want to update us?
I'm still just trying to port over the code to 4.x. So I'm still working on that. Nothing else is really new.
Okay, I'd encourage you to sit down with Giuliano and get some help on that.
Yeah, I'll email him.
Because it can be again, it's one of these things where once you've done it a few times, it's easy, but the first time you do it, it's not so trivial.
Yeah.
And that's not to dismiss that it's hard to learn, but and each model has its own peculiarities about what needs to be done. It's not something that's completely universal. Okay, so just to review what we did last time, one of the things that we talked about was actually just here, which is to remember that when you're displaying chemical fields, you may want to change how you do the display. In the preferences menu, you can select the chemical field that you're going to be talking about, and you can either have a floating bottom and a top range, or fix the top and the bottom. You can do isobars, which can be very useful for figuring out what your chemical field looks like. There's some other options here which typically are not so often used. The other thing which was in the homeworks, and some people seem to have an issue with in some of the homework problems, was to how to actually read values from a chemical field. One thing that you have to be aware of and remember is that the chemical fields are not available as an object by itself. And so you have to do a get concentration field and attach that to a Python object. Now, once you've done that, you can read the values of the field in X, Y, Z coordinates. But you do have to get the field to be able to access it. Once you've done that get, you're all right. So you don't have to keep re-getting the field within a loop, for example, which would be slow. But you do have to make it available within a function. Then we started talking about diffusion times. And this is a very fundamental biological concept. We'll repeatedly come back to this idea of diffusion time, diffusion length, and equilibration time, which are very fundamental biologically meaningful parameters. The physicists will talk about diffusion constants and decay constants, or the mathematicians. But what really matters in terms of the functioning of a molecule are diffusion length, diffusion time, and equilibration time. And so we talked about diffusion time, and there was a comment that molecules can have very, very different diffusion constants, and that diffusion constant determines how long a molecule takes to get from one place to another by diffusion. And the key thing to know is that if you're going from place 1 to place 2 and the distance is L, the time to get from place 1 to place 2, it's random because diffusion is random. But the mean time goes like the square of the distance divided by the diffusion constant. And that means that diffusion slows down very much when you get to long distances. And I give you here some examples. There's this great biomath website that has diffusion constants for lots and lots and lots of molecular species. As I mentioned, there's something called Stokes-Einstein that lets you give a rough estimate of diffusion constant for proteins. It's not a very good estimate, but at least gives you a ballpark. But you could see here, we start out with an ion, a monomolecular ion, has a diffusion of constant of the order of 2,000 microns squared per second. Small molecule like GFP, 7 microns squared per second. And a big molecule like DNA, chromosome, 5 times 10 to the minus 4th microns squared per second. So you have here a factor of 10 to the 3, 10 to the minus 4, so 10 to the 6th range of diffusion constants for typical molecules that you might deal with. And that makes a problem when we're doing computer simulations. We're going to have to face that from time to time. For a typical protein that we're going to deal with, the diffusion constant of 100 microns squared per second is reasonable. We ask, how long does something take to go a millimeter with a diffusion constant of 100 microns squared per second? A millimeter is 1,000 microns. So 10 to the 3 squared, 10 to the 6th microns squared, divided by 100, 10 to the 6th over 10 to the 2, 10 to the 4, 10 to the 4th seconds. So that's a long time. If you think about a human lifetime, you don't want it to take a whole lifetime for a molecule to go from the nucleus of the cell to the outside of the cell. It tells you something about why cells are the size they are. And we also worked on the simple linear gradient. And by simple, I don't mean that it's necessarily something that's easy to learn or something you've seen before, but simple in the sense that the functional form is simple. And one of the things that was actually in the homework that I asked you to do, which we then did in class, was ask you to derive from the diffusion equation the linear gradient. And so if we start out with the diffusion equation, this is in one dimension. The derivative of the concentration with respect to time is d times the second derivative of the concentration with respect to the spatial coordinate, x. If we want to be at equilibrium, that means there are no time changes, so dc by dt is 0. And if I say 0 is equal to d times d squared c over dx squared, I can do the integral once. The first integral says that dc by dx is a constant. The second integral says that c of x is a constant times position plus a different constant. And that was, I believe, homework problem 6. It was homework 6, problem 3.A.
4 point?
No, 4.8,
Yeah.
I think. I'll generally try to avoid heavy-duty mathematics, but that level of math, if it's unfamiliar to you, briefly review the simple first and second order ordinary differential equations. Now, I asked you in that homework problem to start out with no chemical in the cell field, in the field, and have the chemical diffuse in from the left, and to plot what the concentration looked like as a function of time. And I asked the question, how long does it take for that initial zero field to approximate that linear slope that is the equilibrium? And the reason that I asked that question is that the time it takes is given by the diffusion time. And so if I'm going to have the concentration get from the left-hand side here to the right-hand side, the time it takes for a molecule to do that is going to be the square of the distance divided by the diffusion constant. And so if the distance is 100 lattice sites, 100 squared is 10 to the fourth. If the diffusion constant is 1, it means it'll take at least 10 to the fourth time steps to become something that can be approximately linear. In fact, it takes a bit longer than that. It takes 2 or 3 times that. But you can get a rough estimate of the time. If you double the diffusion time, the diffusion constant, the time goes down by a factor of 2. If you double the length, the time goes up by a factor of 4. So the distance is more costly than the diffusion constant is beneficial. Again, that fact that there's a squared term in the time is important. It's not as bad as an NP-complete problem where the time goes up like the exponential of the size rather than the square of the size, but still not ideal. And so here are actually some results from the simulation I did. At time 0, everything was 0. I see, in this case, the concentration is going down. I see it's maximal at the corner. It's still 0 out towards 100. And at 120 Monte Carlo steps, here I've used a diffusion constant of 1. So the distance, x, is about the square root of 120, about 11. If I look at the width of this little band, it's about 10. After 2,500 Monte Carlo steps, square root of 2,500 is 50. And if I look, it's tiny here, but if I look on my x-axis, my blue band is around 50. You can see my curve is not 0 at 50. It goes a little further, but you see it's getting pretty small around 50. Notice this curve is still pretty far from a straight line, right? The equilibrium would be a straight line from the upper left-hand corner to the bottom right-hand corner. Maybe I should have drawn a guide for the eye of the target. Here I'm at 10,000 Monte Carlo steps. The square root of 10,000 is 100. I've got a missing 0 here in my, I've got a typo. This should be the square root of 10,000, not square root of 1,000. So now the chemicals reached all the way to the right-hand side, but as I mentioned, that's still not equilibrated completely. It's not a straight line. If I go to 25,000 Monte Carlo steps, it's getting straighter, but it's still not completely straight. In fact, if I'm going to be mathematically correct, I never reach the equilibrium. I get closer and closer to the equilibrium. I never reach it. But in fact, it's going to take something like 50,000 Monte Carlo steps to be really straight. And so that was that homework problem. I hope people can maybe go back and explore that a little bit more. We also talked a little bit about membranes as domains with different local diffusion coefficients. So this was a problem that I was going to thinking about assigning as an extra homework problem for you, because we didn't really go through it in class in detail. And then I said, okay, people need to work on their projects and they have to catch up. I may give it to you for next week. And we mentioned that cells have membranes, and those membranes partly restrict the movement of chemicals. Sometimes they're very good at it. No DNA will cross an intact cell membrane. Other times they're rather poor at it. Water crosses cell membranes pretty easily. Some ionic species cross, some don't. Things like glucose cross on their own. Cells also have ion channels, which allow other things besides ions through them. And they have pumps. And typically, in our models, we're going to be modeling on a scale where we're not going to model single channels. But we can do that model. And so we did in class an example of this, but we didn't really have time to explore it in a lot of detail. This is a problem that's actually worth doing in quite a bit of detail. I started out by saying to build a wall of frozen, quote, cells. In this case, those cells aren't really representing cells, they're representing membrane. And make it impermeable. That is, make the diffusion constant 0 inside those cells. In that case, if I have a source of a chemical on the left, the chemical comes in, it fills up my region until the concentration on the left is uniform. And that's all that happens. If I allow diffusion across that membrane, but the diffusion constant is smaller than that inside the cell, which is typically true, then what I'll see is that there's a gradient of the chemical. If the membrane weren't here at all, my equilibrium would be a linear concentration gradient. Go from the top left corner to the bottom right, straight line. What's happening here is that I have a wall, a leaky wall. And what that means is that the concentration will stay higher on the left-hand side of the wall than I'd expect. There's a jump to a lower concentration between the inside and outside of the membrane. And then on the outside, the concentration will go down again to reach 0 on the right. And that's something that one can play with. And as you change the ratios of the diffusion constants, you can get quite different patterns. And cells do this. And they're very complicated patterns of concentration near the boundaries of cells because of that. Then I said, let's put in a channel, make a hole in our wall. In this case, the wall is completely impermeable. So the only place the chemical can get through is the hole. And here I've drawn isocontour lines. And what you see is that far away, things are basically parallel. Near the hole, I get essentially concentric circles around that hole. And you'll notice the concentration is lower on the inside near the hole, because you've got essentially like a bathtub. You've got a concentration like you've got a hole like a bathtub that's letting the concentration out. And you'll see the concentration then diffusing on the other side, and you'll get here essentially a circular source of chemical around that hole. If I make the hole bigger, of course, I get more chemical going through for the same gradient. And you can explore what happens as I change the size of the hole, the concentrations, the limits, how many holes I have because they interact with each other. And if I make the membrane partly diffusing as well, I can get relatively complex chemical distributions this way. Connor, go ahead.
So more of a general question, because we've been mostly using CompuCell to study things at like the tissue and kind of like inter-cell level. Does it get used to study like things like, because this strikes me as more of like a singular cell question. Does it get used for that too, or?
Well, the person who just Skyped me and interrupted our class was Gilberto Tomas in Brazil. And he does a lot of simulations of single cells crawling on substrates. So he'll do models where you have an explicit nuclear compartment, an explicit cytoplasmic compartment, a compartment representing the lamellipodium where actin polymerizes and drags the cell around. And he's interested in the statistics of how the cells crawl. And if you want to get those statistics right, you have to have a fair amount of detail. Not individual molecules. You're not modeling individual actin filaments. But nevertheless, you're modeling actin polymerization, depolymerization, the forces it generates inside the cell. So you can actually, it's up to you. We tend to call, I've been sloppy perhaps, I call we tend to call these contour cell objects generalized cells, because really what they are volumes of material. And if you give them the properties of individual cells, then they're a model of a cell. If you give them properties of mitochondria or cell nucleus, then you're modeling subcell components. If you give them properties of a solid, you're modeling a continuous material. If you give them properties of a liquid, you're modeling a fluid. You have the freedom to assign a variety of properties to them. What you'll find is, of course, that if you want to have a lot of spatial detail, so that you're using hundreds of voxels to represent a single cell, then you're going to be stuck modeling a few cells. Maybe not just one, but 10 cells, 20 cells. If you want to model 100,000 cells, which we do sometimes, then you're going to have to make each cell have 5 or 10 voxels in it to make the simulation practical. And that means you're not going to be able to put in that subcellular detail. And so there's always a trade-off between the level of detail and the size in terms of total real-world size that you're representing. But people certainly use it both ways. If you really want to model individual molecules, then there are other software packages that do that probably more effectively, like vCell. Or Simune does that. But you can do quite a bit with CompuCell if you want to model what's going on inside cells. Giulio, in his PhD thesis, did some very nice work modeling the migration of individual molecular species within the cell, both within the cytoplasm and then on the cell membrane. So there are nice models of molecules PIP2 and PIP3. If you're not a biologist, I don't assume those will mean anything to you. They're molecules that are associated with cell membrane, one of which is associated with actin polymerization and cell protrusion, the other one of which is associated with actin depolymerization and cell retraction. And so you can model the reorganization of PIP2 and PIP3 on the membrane, and then that leads to the generation of force that moves the cell. And that's actually a relatively simple simulation. It's one we could do in class. But it's one also where you're going to model a couple of cells, one cell, a few cells. You're not going to model thousands of them. Good question. Any other questions about that? Comments? Okay. So the next thing that we did was our diffusion time assumed that our molecules were immortal. They might appear on one side of our simulation, disappear on the other, but within the simulation, they didn't appear or disappear. And so the next version of our diffusion equation is adding decay, and cells taking up the molecule from the extracellular environment also qualifies as decay. And we'll play with that in a little bit. And that homework problem for diffusion limited uptake, we had that. And the key thing to remember is that decay typically is modeled as what's called first-order decay kinetics, which is otherwise known as exponential decay, which is that the rate of decay is proportional to the amount of the chemical that you've got. And the main advantage of that is that if the amount of the chemical goes to zero, the rate of decay goes to zero, and so your concentrations don't go negative on you. There's some arguments for why that's usually not a bad approximation, and I can refer you to Wikipedia, and you can look up radioactive decay or first-order decay kinetics, and you'd find an explanation. If the destruction of the molecules is independent of each other, you expect that, because it gives you exponential distributions. And last time we took our chemical field equation here, we did the thing that we just did to get the diffusion constant, the diffusion length. I keep saying diffusion length, the diffusion time, setting the time change of the diffusion field to 0, and then hand integrating the diffusion equation. Only this time, instead of saying that the second derivative is 0, we have the second derivative proportional to the concentration. Anytime you have the derivative of something proportional to itself, you're going to either have a sine wave or an exponential. In this case, you have an exponential, and we can write that the concentration then is either a decreasing or increasing exponential of the position. And the key thing here is that the scale factor inside of our exponential goes like the square root of the decay rate divided by the diffusion constant. And that means that when x minus x0 is 1 over this, my concentration is decreased by 1 over E. And so that defines what's called the diffusion length, which is the typical distance a molecule gets before it's absorbed or destroyed. And this goes, so the diffusion length goes like the square root of the diffusion constant divided by the decay constant. And the critical thing here to remember is just the way the diffusion time went as the square of the distance. Here, the diffusion and linearly in 1 over the decay, 1 over the diffusion constant. Here, the diffusion length goes like the square root of the diffusion constant. So if I want my molecule to go twice as far, I have to make the diffusion constant 4 times as big. If I want my molecule to go 10 times as far, my diffusion constant has to be 100 times as big. And the diffusion length is probably the single most important determinant of how biological systems work. It determines the spacing between our blood vessels. It determines the size of cells. It determines almost all length scales in biological problems. We're going to find that for typical proteins, the diffusion lengths range from about 5 microns to about 100 microns. And that means a typical cell is 10 microns. So things that happen within tissues typically happen over the length scale of 10, 5, 10, 20 cell diameters. Distances that are bigger than that, signaling is hard. Distances that are less than that, the signal gets washed out, things are flat. And so that's something that's very important to remember. Again, when we come back to more biological problems, we'll run into it repeatedly. But it's something important to think about here. So I'd like to, since people had some issues doing these hand calculations before, I'd like to bring up a concept which is something that's taught not so much. I'd say physicists do it, but in fact, physicists do it when they do hydrodynamics, fluid dynamics. They tend not to be taught dimensional analysis in other contexts. But I did a numerical, basically algebraic derivation of the diffusion constant by solving the diffusion equation. A dimensional analysis is a way of looking at systems and essentially doing a sanity check. Kind of sanity check that I recommended that you do and ask the question, if you have a function that's a function of the pressure, should it be the function value be large or small when the pressure is small? Should it be large or small when the pressure is large? Dimensional analysis asks you to do this looking only at the units of parameters. And this works when you have a limited number of parameters to play with, a limited number of variables and parameters. So if you want to know what the diffusion length, the diffusion time, and the equilibration time are in your system, and you have a particular equation governing your system, in this case the diffusion equation, the key thing to remember is that there are only certain patterns of units that will give you, in this case, a length, a time, or, and this is a different time, 2 times. And I don't know if people have done a dimensional analysis. Really, any introductory physics course should do it. Often they don't. And this is going to be important because, as I mentioned, especially diffusion length is critical. And when we're going to do simulations, we're going to find that it's critically important to match the diffusion lengths for the species that we're working with. We can often get away with changing the diffusion time or the equilibration time to be non-physical, if that makes our life easier in terms of the time it takes to numerically solve our problem. We have to be aware of making that cheat, but it often helps us. And so let's think about a very simple example. Suppose that we have an object like my pen, and I dropped this object, and I want to know how far it went as a function of time. I can write that the acceleration of my object is proportional to the force. So using Newton's law, F equals ma. The force in a gravitational field is a constant times the mass. And I can say the acceleration, x dx squared by dt squared, d squared x by dt squared, should be careful, is equal to the mass times the force, the gravitational constant. And I need an m on the other side so the mass cancels out. And then I can do the same thing I did before, which says that if the second derivative of something is equal to 0 or constant, then the first derivative is time times that, and doing the integral once more, you get time squared. But let's not do it that way. Let's just do it by looking at the units. The distance I travel here is the initial velocity, which was 0 in this case, plus 1/2 at squared. That's my old result from solving it directly. What about the units? The distance has units of meters, or in our world with cells would be microns. Velocity has units of what? Distance per time, meters per time. Time has units of time, measured in seconds. 1/2 doesn't have any units. Acceleration has what units? Acceleration is the second derivative of position with respect to time. So it has units of distance divided by time squared. And time then has to be squared, because everything has to add up to meters. Let's think about the diffusion equation now. The derivative concentration with respect to time is equal to the diffusion constant times the second derivative concentration with respect to space minus the decay constant times the concentration. And if I have a source term where chemical is coming into my system, call that sigma. Now the units of concentration are amount per volume. And so let's do this out. I have G, grams in this case, grams per meter cubed is sort of an awkward unit. Nobody would actually measure something in that, but if I'm doing biology, it would probably be moles, micromoles per micron cubed or something like that. If we're doing dimensional analysis, we don't care what the units, which unit we're using for length or mass or anything else. We do care about that we have one. So we now have C has units, concentration has units of grams per meter cubed. Time has units of seconds. And those square brackets, if you're a chemist, you're used to those square brackets representing concentration. There's another use, which means that square brackets represent the units of the thing that there's inside the square bracket. And of course, if it's Python, the square brackets are a list. So we have to remember the context in which we're using our square brackets. And on the right-hand side, we have D, the diffusion constant, times G over M cubed for concentration, divided by distance squared for the derivative, minus gamma times the concentration, plus the secretion rate. And so you now have a little exercise. I've talked a lot. So your exercise, and I'll give you a few minutes to do it, is find the units. Yes, closed intervals too, thank you. Yes, well, we run out of symbols pretty fast, I'm afraid, especially for brackets. It's bad enough in Python having three kinds of brackets. Square brackets for lists, curly brackets for dictionaries, and regular parentheses for functions. So I'm going to ask you now, you see this picture, this equation, it has to be consistent. What are the units of D? What are the units of gamma? And what are the units of sigma? So why doesn't everybody take a few minutes and see if you can figure that out? Is the question clear? If you're stuck, that's fine. Just if you want to think about it more, that's fine too. Either way. Maybe we should do one and give people a little bit of a hint. So I think this must be Giuliano's slide, right?
Yes, it is.
Stuff per volume, it sounds like Giuliano. So let's take the sigma, the rate of secretion. What are the units of sigma?
So if I'm understanding this method right, the way that it works is that we basically need to get everything to have the unit structure of what's on the equal sign, which means everything kind of encapsulated within a minus or plus sign. So like we're basically getting the multiples to come out. So sigma should have just literally the same unit structure as the left-hand side of the equation.
That's right. So that's grams per meter cubed per second, or concentration per second. That's the secretion rate. And I guess there's the old saying that you can't add apples and oranges. If you're adding two things, they have to be of the same type. That's true in computing too. We can't. Python will let you add between almost any two objects classes, provided that it's the same kind of object on either side of the equal sign. I mean, either side of the plus sign. If I try to add a string and a number, it'll get confused. Now, of course, I can multiply a string and a number in Python, but I can't add them. So yes, so sigma has units of grams per meters cubed per second. Okay, what about gamma?
I think gamma will be just something over seconds, right? So you'll have this multiplying, it will have the same thing. Like 1 over seconds.
1 over seconds. Okay. The one that's a little bit more interesting is D. What about D?
That's where I'm stuck.
Well,
So I think basically like going through it, like first of all, we need to remove the meter squared from the bottom. And we also need to get seconds on the bottom. So it threw me off visually to see it like in the numerator, but that's the same as like multiplying it to the left. So I think it should be meter squared over second.
well, let's see. If I start out here, I want D equals something. So the units of D equal something. I divide through by G over M cubed. That cancels that. So then I have 1 over seconds. I multiply through by M meters squared. I get meters squared per second equals D. So that's the answer. So all I'm trying to do is put my unit, the thing I'm trying to find the units for on the right by itself, and whatever is left on the left is my answer. In the case of sigma, I don't have to do anything because it's not multiplied by anything on the right. So it's just sigma equals that. In the case of gamma, I have to divide through by G over M cubed. So I'm left with 1 over seconds. In this case, I have to divide through by G over M cubed, 1 over seconds, to multiply through by meters squared. The units of D are meters squared per second, or length squared per second. In our case, we measure things in microns squared per second rather than meters squared per second. Diffusion constant of meters squared per second, I don't know whether there's anything in the universe that has a diffusion constant. I guess in the star where the temperatures are incredibly hot, you might get diffusion constants that high. In our world, nothing has a diffusion constant that high. Okay, does that work for people now? Does that help? And I understand that it's not immediately, this isn't something that comes naturally. But you see, it's also not very hard once you figured out what you have to do. You're just multiplying things by simple. Okay. So here I'm doing that again. Sigma is not multiplied by anything, so you just get the result. For D, I have to multiply through by divide through by G over M cubed, multiply through by M squared. And for gamma, I have to divide through by G over M cubed, left with 1 over seconds. Okay. So now I want you to derive the diffusion length. And so I splashed the result. You've already seen the diffusion length, the answer, but let's come back to that. I want to know, looking at this equation, how far a chemical can get. And if it's a diffusion length, and I've already told you diffusion length depends on 2 things. It depends on diffusion, which is this first term, and it depends on decay, which is the second term. And so what you have to do is think about how can I combine the diffusion constant D and the decay constant gamma to get a length. There's only one way you can do it. And you have the units here. Remember, the units of D are length squared per second. The units of decay are per second. How can I combine D and gamma to get units of length? And I guess I really should have had these results on the next slide. I'm sorry I left them out. So you want to get the diffusion length as a function of D and gamma. And there's no secret. Diffusion length has units of length, meters in this case.
Doesn't that mean we're dividing d by gamma and taking the square root to leave it with just n?
That's the answer, yes. It's the only way to do it. Because the only thing here with d and gamma, the only thing that has length in it is d. So you need a d, otherwise you're toast. You don't want seconds. The only way you can get rid of seconds would be to divide d by gamma. That gets rid of the seconds. That leaves me with length squared. I want length, so I have to take a square root. So you've just solved that problem very nicely. And so instead of having to do the integration of a differential equation, I look at the units and I can write down the diffusion length. So for people who haven't remember their calculus, this is a way of avoiding doing the calculus. This is arithmetic. It's not any more than simple algebra and arithmetic. So that much I think I will demand of my students. Okay. Okay. What about the time the field takes to forget its initial configuration, the equilibration time? Again, I should probably come back here to the things that I have available to me. I have the secretion rate, the diffusion constant, and the decay rate. And I want a time. Maybe I can give people a little hint to get started. Let's think about the secretion rate. Can the secretion rate show up in the equilibration time? If I look at the units of secretion rate, I have mass divided by meters cubed times time. Well, I want something with time in it. That's all right. But do I want mass in my result? No. Is there anything else that I have that has units of mass? No. So is there any way I can cancel out that unit of mass when I combine things? No. So sigma cannot appear in the equilibration time. Can't be there. So somebody tell me now, can when I look at the diffusion constant, can the diffusion constant show up in the equilibration time?
No.
Who's talking? Jackson? Good.
Yeah.
Why not?
When I went about it, I was just trying to find the easiest way to get to just seconds. And I'd have to go
Okay.
through and figure out a way to get rid of the meters squared.
Right. Is there any way to do it?
Not that I could think of.
Right. You have three things to play with: sigma, d, and gamma. The only one that has meters squared are in sigma, which I'm not allowed to use, in d, and in gamma. I've ruled out sigma. That leaves me with d and gamma. There's no way to combine d and gamma that gets rid of the meters squared. So I don't have d either. All right. So I'm left with gamma. So if I want to get a time out of gamma, what do I have to do?
I did 1 divided by gamma.
1 over gamma.
Yeah.
So the time, the equilibration time of the diffusive system is 1 over the decay rate. That's it. I think people may be stuck because it's such a simple answer. Sometimes the simplest things are the most confusing. Once you see them, they're easy, but they're slippery because they're so simple. But you see how the thinking goes. You look at each term, you say, can I combine them to get the thing I'm looking for? In this case, we could rule out sigma, and we could rule out D. And the thing we were left with was what we had to have. That's it. Now, this idea of equilibration time is useful because this is how long it takes for the system to stabilize. And you'll notice that if there's no decay in our system, the system in principle takes an infinite amount of time to stabilize. It never forgets its original state. And since our original state typically is something artificial, like making everything uniform or having nothing of something, that initial state that we have is often not something we really want to be present in our long-term behavior of our system. And so typically we will turn on some small amount of decay in our diffusion equation to get rid of that artificial initial state. Otherwise, we have very sensitive dependence on our initial conditions. Okay. The last one is just a bit trickier. And it says, suppose that we wanted to figure out the diffusion time. We know the answer already. But let's do it this way. We want to know the time it takes for a molecule to go from point A to point B. What is that time? And we can come back to our parameters, sigma, d, and gamma. And we want to know what that time is. Now, when you're thinking about this problem, you might want to start out by thinking about two things. One, that I showed you that derivation before we talked about decay. And two, to ask the question, is there some other unit, something else with units in this problem besides those three parameters? What did I say that we have in the problem? I said, how long does it take to get from point A to point B? What do I know about point A and point B? Somebody.
Just their locations that you're referring to? So the
Right.
distance between them?
So I know their distance. What's the unit of distance in this world?
Meters.
Meters. So I have L, which is the distance, and it has units of meters. So I have an extra, in this case, a variable, extra parameter to play with, which has units of meters.
And just a question, L is defined as the square root of D over gamma, right? That's what we saw before.
That's what you were supposed to be deriving, right? No, L is the, I can call it something else. Call it R, the distance between the start and the finish point, the starting point and the finish point. I want to know how long does it take to go a distance R? Although I think I used the symbol L in my slide. Okay, well, let's think about this together. If somebody has the answer, that's fine too. But let's do the same argument we did before. Can I have sigma in the time?
Mm.
No, because I can't get rid of my mass. What about gamma?
No.
It has units of time.
Yeah, it just doesn't help get rid of the M.
That's right. Doesn't help get rid of the M. In fact, it would be worse because if I use it with the M, it'll get rid of the time, which is what I want. So now I'm left with D. So if I want to get D to give me a time, what do I have to do? I want to combine D and L to get units of time. What do I have to do?
Would you square L and divide by D?
That would work. So let me clear my, where's my annotator? That's exactly right. So I have so I'm asking for the diffusion language. So, okay, the time it takes to go from A to B, the distance here, I called it dt instead of L, is got units of M. As you say, distance squared divided by D gives me units of time. So L squared, or distance squared, divided by D. That's how long it takes to go. And that was what we had before. So I realize that if you're not used to doing this, it's tricky. And if you have lots of things with the same units, suppose I had 10 different lengths in my problem, then I wouldn't necessarily know which one to use. And so that can cause an issue. But in these simple equations, structural like diffusion equation, you can do a lot by arguing just on what's called scaling. And this kind of scaling is used in biology a lot. You'll run into something called allometric scaling, which is I make a measurement in a mouse, and I want to try to guess what that means for a human being. And I notice that the mouse is smaller than a human being. And so if I've made a measurement in a mouse heart or a mouse lung or a mouse liver, I have to take into account the fact that the volume of that mouse heart, lung, or liver is much smaller than that in a larger person. And therefore, I have to think about whether that length goes in linearly, quadratically, or as the cube. Does the thing depend on the volume, on the distance, on the area? And I'll find that some things change in interesting ways and some don't. The weight of the animal goes roughly like the cube of the size. The heart rate of the animal goes up as the animal gets smaller. The mass of the brain scales in a funny way. It increases with the size of the animal, but not with the mass, not as fast as the mass. Our brains are smaller relative to our bodies, than you would expect if you took a mouse and scaled it up to our size. Our metabolic rates are slower than you'd expect if you took a mouse and scaled it up to our size. And there's a whole discipline of scaling relations, which asks the question, as an animal gets bigger, how long does it live? Bigger animals tend to live longer. As an animal gets bigger, how much food does it have to eat per day? Bigger animals eat more food, but they eat less per unit mass. And then there always are exceptions. There's some animal, you get this nice line, whether you have one variable like the size on the x-axis and whatever it is you're measuring on the right. There's this line. Do this in log scale to get the exponent. And then there'll be a few outliers. So in general, smaller animals live a long time. It's a short time, and large animals live a long time. But naked mole rats are small and they live a long time. Horses are large and they live a relatively short time compared to their size. Our brains are bigger than we would expect given how much we weigh. Dinosaurs' brains were smaller than you would expect given what they weighed. Whales have big brains. Yes, the comment is a good one. There's the rough estimate that Giuliano says. There's a rough estimate for mammals. You would say 2.5 billion, approximately. Say a billion heartbeats is about right. Give a factor of 2 for. Most mammals have a lifetime on the order of 1 billion heartbeats. Next time you're counting your pulse, you can count down that way.
Definitely an eerie way of counting down your days.
Well, so in a sense, it makes sense.
I did that for a deadline once. I calibrated a ticking clock timer to my average heart rate with the joke that as I became more panicked about the deadline, I would get more time because my heart rate would be increasing.
Well, our perception of time is very fluid, and so that's not an unreasonable thing. Yes, heart effects. That's a good one. Yes. So there are some beautiful books on these kinds of allometric scaling relations. Often they give you meaningful results. Occasionally they don't work. Again, it typically they don't work when there are multiple length scales to choose from or multiple time scales to choose from. In principle, if you have 10 lengths, you can make all sorts of different combinations of length. And so you don't, that's when this kind of analysis fails. But it's a good, just the way thinking about what happens in the limits of small values and large values, and does it make sense, is a good sanity check. Checking that the units make sense is a good sanity check. If you because Python will allow you to add if you have concentrations in X is concentration of molecule 1, Y is concentration of molecule 2. Python will allow you to write Z equals X plus Y squared. It's perfectly good algebra because the units are not represented and they're not checked. But biologically, physically, that's a meaningless thing to do. And so checking consistency of units is something that's quite powerful as a way of catching mistakes. And one problem with CompuCell compared to some packages is that it doesn't have a built-in concept of units. We have to do that by hand. Okay. So I hope that exercise was at least a little bit useful. The next problem was a homework problem. And I can go over it in a variety of levels of detail depending on how people felt about it. And this was to write, last time we did what we called the linear gradient. where we had no decay. We had a source and a sink and no decay. In this problem, we turn the decay constant on and we ask, how does the system change? And I wanted there was a homework problem, and so if everybody did it and was successful with it, then I don't need to assign it here. But if people had issues with it or want to talk about it more, we can do it together. What do you think? I'll tell you what, since we're over our half time, why don't we take a 5-minute break? And when we come back at 7 o'clock, you can tell me whether you want to do this problem or not. Okay? How's that? Okay, so I'll see you back at 5 minutes at 7 o'clock. Okay. So, if people would like, they could show me the answer if they've already got it. Otherwise, we could work on it together. What do you think?
I'm going to request working on it together.
Okay. Does anybody have the answer that they'd like to show? Maybe more exciting than my doing it. Maybe we should do it on somebody else's screen and talk them through it. What do you think?
That's fun. I can volunteer for that.
Okay. So the only problem with that is that that I can't screen share the slide at the same time if you're doing it. But that's fine. Let's think about this. So what I want people to do is create a chemical field. Here I called it oxygen, or you could call it anything you like. And I want you to set the global diffusion constant to be 1 and give it a small decay constant. Here, a factor of 1 over 1,000. And then I've said impose a constant concentration on the left, 0 concentration on the right. And then what's the shape of the result? Do people have the code from last week where we measured the concentration as a function of position? If you do, that's useful. If you don't, we don't absolutely need it. So why don't everybody fire up CompuCell in one form or another and see if you can remember how to do this part of it, and then we'll walk through the rest together, okay?
I'm trying to remember which number this was in the homework. Was this 4B? I actually, let me double check. I think I have working code for this one, so I might be able to show it. I need to restart my CompuCell though. It's been a little buggy. Give me 2 seconds.
Well, maybe I should have a show of hands. How many people were able to get this to work the way they wanted to? Connor was. Josh was. Joel, were you able to get through it? No. Jackson? Andrew?
I tried it.
Okay, so in that case, I think that those of you who've done it successfully, help out your classmates, and let's do it together.
Okay, yeah, I have a working version. Would you like me to make a new one from scratch though, or just walk through the one
I think probably let's create it. I mean, watching me code is boring, but let's watch you code. So let's use Wizard to create a new simulation.
Okay, here, let me.
Not necessarily to do the whole walking through the calculate and plotting it, but let's just get the chemical field together.
Okay. Let me share. I can guarantee I'll make enough mistakes that you'll feel better about your live coding. Can everyone see my Twit now? Okay. So new project. Let's call it homework 6 or 2. I think it's 100 by 100.
That's fine. It's not critically important.
Okay. I'm just going from memory. Evan, that doesn't matter because we're going to get rid of that. Okay. Nothing here for this one. Oxygen. Skip over that stuff. Okay. All right, spirits of CompuCell 3D, Zoom people, it is your turn to guide me.
So somebody, where does the diffusion solver live?
In the XML.
Okay, so maybe let's start looking at the XML.
So I'm going to go ahead and get rid of this. I'll just comment it out. So that's done now. What do people want me to do next? Come on, Hive Mind, I'm powerless without you. I can't take action.
I'll put in a reminder because I didn't give the parameter values that they're off the screen because I can't share them at the same time, but I'll put them in the chat.
Set the boundary conditions.
Okay. So let's see. So I need to. Oh goodness, Zoom is gonna block my part of my quit bar. Yeah, let me. There we go. There's supposed to be periodic boundary.
I think in this
conditions.
one we want constant value on the X. So constant
That's
value.
right. That's right. Okay. And then it was
Periodic on Y.
Thank you. Thank you.
And I noticed in the homework that people often had trouble remembering. They'd control one boundary, but forget that there were 4 that had to be done. And so it's easy to get one direction off, and then you get something that looks funny. Okay, that's good. And what were the values that were supposed to be on the left and the right?
I think this one was this one 1 and 0? I'm going from memory right now. Someone can correct me.
Well, 1 or 10 on the left here don't make a big difference, but on the right it matters. So I think you want 0 on the right.
Yeah.
Okay. And that will run. So why don't you try running it?
Alrighty.
In the famous last words department, right? That will run.
That's what I think about all of my code. All right, let's tile and.
I think you're not screen sharing the player.
Oh my goodness. Okay. You'd think after all these years of using Zoom, I would finally get that right. All right. Well, spoiler alert, it does run. So let's. Oh, sorry. My CompuCell has been doing this weird thing where it goes back to loading a previous file. And I sometimes need to close out of it and try again. So let me go ahead and do that.
Are you on a Mac?
Nope.
Yep, sometimes it does with me too.
Huh.
I have to
Um,
reopen the phone. Yeah.
Yeah, I was considering it's only started recently. I was considering letting you know, but I wasn't sure if it was a my computer issue. So I was going to try debugging that first. But
No, please let us know. That's a
Okay.
bug.
Okay. So I can tell you more about how that's been happening later. But for now, here's our chemical gradient. People can see it now?
Mm-hmm.
Okay.
So there's something that we discussed in class that you might want to do. What?
I'm going to mute for a second. There's a train going by.
I don't know why your max distribution is going nowhere. Hold on a
Yeah,
second. Let me try something.
I'm a little, I don't think it does that in my other simulation.
Well, so you might want to change the color bars so that you could see the gradient. And you might put the ISO value. Well, it looks like you've got the ISO values turned
Yeah.
on.
I have those on. Okay. Okay, and that makes sense because we set the max for 10 before. So the difference between this and my other code is I set the max for 1, which is why I was able to keep it to the 1 to 0 range. So that's why it looked off. Okay, that makes sense.
Okay, great. And so now the other thing you might want to do is the instructions were use a D of 1, diffusion constant of 1, and a decay constant of 1 over 1,000.
Go back to Twitter.
You'll have to share the Twitter.
Yeah. So let's see. Here, what was it? It was
1 and 1 over 1,000.
okay. Wait. See if player opens up the right thing if I stop and go. I'll share again in 1 second. Okay, that seems to have worked. I did something horribly wrong in the past couple seconds, apparently. That doesn't look right.
My guess is that it's not reading the 1 over 1,000 properly.
Okay.
It looks to me like it may actually be interpreting 1 over 1,000 as 1,000.
I see.
So you probably have to do 1 e to the minus 3, e minus 3.
Okay.
There are some places in CompuCell SCC3DML where it does math interpretation, but not everywhere.
That's good to know.
And I actually would have expected 1 over 1,000 to work, so I'm a bit surprised it didn't. Okay. All right. So that looks great.
Yeah.
So I guess the one other thing, I think you have code written that will actually plot the values, right?
Yeah. Do you want me
So
to pull that up?
why don't you show people the code, and then instead of typing it through, why don't you paste it in? And so,
Okay.
is everybody able to follow what Connor's done? Maybe we should give people, while you're doing that, why don't we give people a minute to replicate what you've done? Okay, I think let's give everybody a minute to catch up.
Do you want me to paste the code for the plot?
That would be fine if you like. If everybody's caught up, we can keep going. Problem with the code for the plot always is it has to, there are two plot, that some of it goes in the start, some of it goes in the steps, so there are two places.
I don't think it pasted everything. I think it cropped. Let's see.
Yeah, Zoom has a
Character limit.
such.
Mm.
Yep.
Yeah, just be careful with indentation now, because it's out of format.
Okay. Connor, why don't we come back and see what you've got?
Let me go ahead and also I just for your information, Professor Glazier, I just had the bug again where I try to share my new code. And it shares a previous one. So I'll try to pay more attention to when that happens so I can see if there's a specific like process that I'm doing that makes it lead to that. But for now, all I know is that it's doing it
Isn't-
semi-randomly.
When you close, do you have the habit of closing CCT3 every time that you're going to open a new?
You mean like each time I'm going to open up a new project? No, I don't do that.
Okay.
Because typically I'll be working on multiple projects in parallel. I tend to work a little bit sporadically depending on what's going on. Also, just a disclaimer in advance, the code that I ran before where I actually had the line moving, I had the global decay constant set to 0, so I just changed that.
Great.
So I haven't ran it. But basically, if we go to the plotting, I basically did it the way that we went over in class last time, where here I'm erasing all data at each step. So instead of accumulating the points over time, it's just basically going to plot. So I'm centered along the y-axis. So it's looking at all those x coordinates going straight across the middle, and then it's plotting that line as I go. And then the rest of it is just kind of like the basic import functions. So I don't know if people want me to stay on this for a moment or if people want me to go ahead and run it. Also, always welcoming critiques of my programming style. If I'm doing anything sloppy, will not take offense if you want to tell me that I could have done something better. Okay, I can go ahead and run it if people are done just staring at the code.
People should feel free to break in and ask questions. At some point, you're going to have to do the same thing. So Connor's being very nice and being the guinea pig for doing the development.
There's going to be a delay in my development because I'm going to have to close fully out of CC3D. It's opening the 4.2, the one that I did without the line, repeatedly. So
how are you opening? How are you opening it?
So and that's true. This might just be a user error. So what I'm doing when I want to switch projects, instead of hitting the stop and go again, I'm going to the file that I just saved and then intuit it saying open in player. And then it's saying, do you want to run this file when this simulations are open? I'm saying yes. And then it's giving me the old simulation, even though I said yes.
So I'm puzzled by that, but
it genuinely could be me. I might be doing something arbitrary that I'm missing.
no, I don't think so. I mean, if so you're highlighting the name of the file that you want to open, You're right-clicking on it, and then you're saying open in player, and it's giving you the wrong file.
So I'm highlighting the one that I want to play, and then I'm going
Why
to.
don't we see that? Let's do that
Sure.
together.
Sure. Okay. So
I'm sorry, that's really not educational, but it's educational for me and Giuliano to see what's going on.
okay, fair enough. As long as everyone's getting an educational experience. So I'm highlighting. So this is the one I wrote before, which was playing.
Right.
Now I want to play my original homework 6-4, and then I'm going to project and hitting open in
Okay, so
player.
I don't know that will work.
Okay, that's what I've been doing.
So
What should
I
do
would
instead?
highlight in the left hand navigation in the tree hierarchy and the left hand
Over here?
bar. Yeah. Highlight the one you want to open.
Okay.
Right click on that.
Okay.
Now say open in player.
Okay. So now I will go to player. Can you all see my player right now, or do I need to reshare?
No,
Okay.
sure.
Resharing. So fingers crossed, it's saying current simulation. Do you want to stop it immediately, run the new one? I'm saying yes. Go ahead, initial. Okay, so that works. So it just doesn't work when I highlight in the top bar. I need to go to the bar on the left. That's good to know.
So what's happening is whatever's highlighted there is the thing it will open.
Okay.
And so you could switch between those tabs without switching which simulation is connected to player.
Okay. Okay.
And
Gotcha.
I agree that that's a little bit confusing, but you might want to be able to edit pieces of a code without switching which one is being targeted. So that's
No, that makes sense. Okay. All right. Well, moment of truth. Okay. Yeah. So here it's running across, and I can run it again, but we basically see that linear relationship forming.
Which is strange, given that we have decay.
That doesn't have decay, yeah.
Yeah.
Right?
So that was why. So let me go back to my.
You can do this one by hand. So you could go into the steppable, the diffusion solver steppable under model editor and change those parameters on the fly. So if you keep it, leave it running, go in there. You have to open everything up. It's sort of a hierarchy by then. Okay. No, it does have the global decay set to 10 to the minus 2. So the answer here is that, the diffusion constant is 10. So what's the diffusion length? Remember, what was the diffusion length?
That was the So is the issue that that I have like the linear relationship is because I have it at?
Well, there's nothing wrong with so the next exercise was actually to play with the diffusion constant and the decay constant and see what happens. In particular, there's a question of equilibration time. Remember, the equilibration time is set by the decay constant by itself. The diffusion length is set by the ratio of the diffusion constant to the decay constant, square root. So let's say what the diffusion length is. The diffusion length, remember, is d divided by gamma square root. So what's d divided by gamma? 10 divided by 10 to the minus 3, 10 to the 4th.
Oh, okay.
The diffusion square root of that's 100. So the diffusion length here is 100.
Okay, that makes sense.
The difference between the linear function, the straight line, and the exponential is not that big. It's there. If you keep running this, it won't ever get quite straight.
Okay.
But why don't you make the diffusion constant 1 instead of 10? See what happens. Now you can hit run and it'll keep going.
Okay, that makes
sense. Now you see the exponential. Notice that it's taking time to equilibrate. Look at the time bar. For 10 to the minus 3, you expect it to take 1,000 time steps to equilibrate when you make a change. Well, you're at 2,000 time steps now. So why don't you hit pause? And let's change the global diffusion constant to something else It asks how long does it take to equilibrate. You're on about 3,000 time steps now. So let's why don't you make the global decay constant? Somebody want to vote bigger or smaller? The diffusion constant, bigger or smaller?
Let's go small. I want to see that line touching the
Okay.
bottom.
How
Okay.
small? Give me a number. What do you want? Like 0.2?
Sure.
Okay. And now let's look. 3,000.
So in this situation, since we didn't,
4,000. So now you're at 4,000, well, almost at 4,000. So you should be equilibrating around
Sure.
now. Still going down a bit, but it should take on the order of 1,000, 2,000 time steps to equilibrate, which it does.
so in this situation, each Monte Carlo step is 1 second or 1 unit of time in this case, since we didn't establish anything.
Just one our time units are Monte Carlo steps.
Yep.
So instead of
Okay.
seconds, you have MCS. And our length is measured in voxels rather than meters. But, okay. So now you could ask the question, suppose I multiply the global diffusion constant by 10 and the global decay constant by 10. What will happen to the curve? If I multiply them both simultaneously.
Are you asking people to pause and think about it? Or are you asking me to do it so we can? Okay.
I'm asking you to think about it first.
Okay.
Wouldn't the decay affect more? So instead of we seeing the same thing, we'll see a reduction. Like the line will
Okay.
go down.
So you think the line will go down if I multiply both by 10. Anybody else have an opinion? How many people agree with Joel? You could do a show of hands up, down, if you want, with the icons. I want everybody to vote before we go on. Joel says that it will keep going down. Josh says it'll keep going down. Anybody else? Jackson,
Oh.
yes? Okay. Andrew? Yes, everybody says it'll go down. Okay, let's try that.
We all believe in you,
multiply,
Joel.
multiply it by 10, make it
Please
2,
don't. Please.
and make the
Sounds
global
safe.
decay constant 10 to the minus 2.
Yep.
Okay, now run it.
Okay.
God bless.
Remember, I told you the most important thing biologically is the diffusion length. If I multiply the decay constant and the diffusion constant by the same factor, what happens to the diffusion length? What was the diffusion length? It was the square root of d over gamma. So if I multiply both by the same thing, the diffusion length doesn't change. What does change if I change both together? What happens at equilibration time?
Should equilibrate.
Equilibration
Faster,
time
right?
was 1 over the decay constant. So if I make the diffusion constant and the decay constant bigger, it equilibrates faster, but I get the same final result. So that's a trick we'll use in numerics, which is biologically we often don't care about the equilibration time. We want it to be fast because we don't want to sit around waiting for the simulation. But we do care about the diffusion length. So we calculate the diffusion length first because that's given by the biology. And then we say, well, we want it to equilibrate in 100 Monte Carlo steps. It means our decay rate has to be 1 over 100. And then that tells us what our diffusion constant has to be to get the real diffusion, the diffusion length we want.
So the last adjustments we saw in the line it was because it was faster. It was happening faster. It would happen. It would just take a little bit longer. And now it was
Right.
faster.
We were running slower, so it hadn't completely equilibrated.
Got it.
So now it's equilibrated. Why don't we multiply by 10 again? Multiply both by 10. Well, you multiplied 1 by 10. Okay,
Yeah,
good.
sorry, my finger slipped.
No worries. That's actually
well.
interesting. See how fast that equilibrated now?
Yeah, that was an intentional example. I wanted y'all to see how quick it went.
Because now you have a decay constant of 0.1, so it takes only 10 Monte-Carlo steps to equilibrate. And we could do the opposite. Let's divide everything by 1,000. It's running. Nothing's
Yeah, it's
changed.
just right, because that relative relationship's still
Yeah.
the same.
Okay. So that was that now I think that was a very thorough examination of that homework problem. So I want to thank you for doing that. And, maybe you could zip that student materials folder. I think everybody can use. We could share that code if people. Does anybody else have any comments on that problem? I think it was nice to see it solved so completely. And again, I think I understand it takes time to develop a sense of what makes sense, what doesn't, how to work through these problems. It's not trivial to do. Okay, so here was my solution. You see, no decay, straight line, put decay in, you get exponential. So now I'd like to switch talk a little bit about sources and sinks of chemicals. And at the rate we're going to be talking about sinks, but not sources. And the dealing with sources and sinks is a little bit more complicated. And we're going to find that there's some numerical approximations that mean that we're not perfectly solving the diffusion equation all the time. And so that now we're going to include the idea of, we've already seen this, a decay, we had a decay term. The decay term is proportional to concentration. A sink, in principle, can take material out at a rate that's not linear in the concentration. And the source can put material in at any rate it likes. And I guess that my notation is not entirely consistent from slide to slide, because some of these slides are pretty old. If I think about secretion biologically, there are a lot of different ways that molecules can get out of cells. And we saw already in the example of ion channels, and just the membrane itself, a molecule can, in principle, diffuse directly across the cell membrane. Depending on what molecular species it is, that diffusion could be very slow, but it's possible. Maybe not for something like DNA, which is big, but for small molecules, yes. We also saw ion channels. Ion channels can allow small molecules through. Pumps can actively move molecules across the boundaries of cells. Cells also have pathways called exocytosis. Those show up also in viral synthesis and release. So cells will actually make, coming off of the Golgi apparatus, vesicles, membrane-bound vesicles, which are full of particular chemical species. Those vesicles can then fuse with the cell membrane and release chemicals to the outside world. A classic example of that would be in synapses, where the transmission of signals across from cell to cell, from axon to dendrite, is done chemically by secretion of vesicles, which release a molecule that then binds to receptors on the opposite cell. And there are a number of these pathways. We're going to typically ignore all of the details of how secretion happens biologically. If we need it, we will begin to think about it. But in general, we will assume that molecules disappear from inside the cell and appear on the outside of the cell. Secretion in CompuCell has a wide variety of possibilities. And in one of the homework problems, well, that's specific on absorption, we had a whole series of options called uptake, total count of various kinds, some of which were a little bit confusing. Secretion in CompuCell has also gone through a variety of iterations, some of which are in the separate secretion plugin, some of which are in the diffusion solver, and some of which live in Python. And we need to decide how we're going to do secretion if we're going to do it. They don't all behave, they should behave numerically identically. They don't all behave quite identically numerically. There are questions about when execution happens that may make them give slightly different answers. But if we're going to control secretion from plugin, from Python, we still need to say we're using it. And our principle that nothing exists unless we assert it. So the first thing that we have to think about is where secretion is happening. It's going to be true for uptake as well. And this is ultimately going to be a biological problem or physical problem, which is where does secretion actually make sense? In many cases, the primary limitation of secretion or uptake is the time it takes the molecule to cross the cell boundary, the membrane. In that case, we might care, and here I keep saying secretion. The slide deck is not quite what I thought it was at this point, because I keep talking about absorption, and the heading is secretion. I should fix that. I'm not sure how that got messed up. I apologize. But it's going to be true for secretion and absorption. Both of these are going to be true. We can secrete just inside a boundary. We can secrete just outside a boundary. We can secrete everywhere in the cell. We can secrete at the center of mass of a cell. And we're going to be able to keep track of how much is secreted or absorbed with the syntax total amount or total count. The other thing that we're going to be able to do, and again, some of these slides may be a little bit not quite in sync with each other, is that in the case of things like epithelial layers, the layers of cells that line your gut or your skin or your nose, there's one surface of the cell, the top surface, the apical surface, that's responsible primarily for secretion and absorption, at least of certain species. And so we're going to find that it's quite useful to restrict secretion or absorption to surfaces of cells that are in contact with a specific target. So we will secrete on contact with medium or absorb on contact with medium. Now there are going to be situations where we still need there are going to be situations in which we need more detail, and then we'll have to do things like compartmental cells. But we very well might want to have a layer of cells in a line, like the one I've shown here, where the molecular molecules, the way molecules pass from cell to cell horizontally is fundamentally different from the way molecules pass vertically from the cell to the extracellular medium. In particular, there might be things like gap junctions, which are holes between the neighboring cells this way, but there might be active pumping exocytosis or endocytosis of vesicles carrying molecules either in or out of the cell to the medium. Secretory cells are highly polarized. You'll see they're designed to make material, transport it to the top surface of the cell, and then release it into the environment. And they release on the top surface only. There are other cells that pull material in only from the top surface. And so this asymmetry is something that we need to anticipate because implementing it by hand is pretty complicated. But anticipating that variety of possibilities has the downside that we have a lot of options in CompuCell that we have to be aware of. And as somebody found out, the older version of CompuCell that's up on NanoHub doesn't have all of those possibilities enumerated in the TWIDIT. They all exist, but you have to type some of them by hand. Okay. We're also going to have this idea of tracking amount, and there'll be this total count option that will exist for both secretion and absorption. And some of those commands get rather verbose. Secrete outside cell at boundary total count. They get long because of all of the permutations. Okay, if we're absorbing, and this keeps saying secretion, but then the text is about absorption, so I will talk about absorption. If we're absorbing a molecule that's diffusing, like glucose, the glucose is used inside the cell, and the primary block, as I mentioned, is the membrane. And so in that case, we basically, once the glucose gets into the cell, it's used immediately. And so the absorption happens just inside the cell membrane. In another case, which is pretty common, there could be receptors which stick out of the cell and bind molecules that are circulating. Those receptors could be doing something useful, like binding a growth factor, or they could be doing something less useful, like binding a virus and bringing it inside the cell. You've heard of I'm sure by now, the ACE2 receptor, which is one of the primary ways that SARS-CoV-2 gets into cells. It's not that there was any evolution of the ACE2 receptor that's to bind SARS. It's the other way around. But that's a receptor that exists to receive a molecule that regulates blood pressure. That happens to bind, unfortunately, spike protein on SARS-CoV-2. So in that case, when there actually is a set of surface molecules on the outside of the cell, the uptake is happening just outside the cell. And that's why you'll find that there's this option of inside cell boundary and outside cell boundary as possibilities. Another thing that could happen is that we could be imagining that our cell, we're looking, instead of in cross-section at a layer of cells, we could be looking from the top. And if we're looking from the top, essentially every point on the cell could be pulling material in or secreting it, because there's uniform secretion from that point of view. If we take a section, we're only coming from the surface. If we look at it from the top, we want every place in the cell to do it. And so we could imagine that we secrete inside or absorb inside the cell over the whole cell area. And then, because these kinds of things can be slow, and because packages like Physicell, which you might want to replicate, don't have all of these options, we might want to imagine that our molecule is either absorbed or secreted at the center of the cell. And that has some peculiarities because you get very spiky distributions, spatial distributions. But it's also not totally unreasonable. If there's a molecule that comes into the nucleus and binds to the DNA, to assume that that molecule is used up when it gets to the center of the cell, if we imagine the nucleus at the center of the cell, is not totally wrong. And so there's going to be an option called inside cell at center of mass. And so we're going to always have these options, which are where the secretion and absorption is happening, and also the possibility that we only secrete or absorb on contact with particular types. Usually that will be type cell to medium, but potentially if the cell's sitting on a substrate, like an extracellular matrix, it could be secreting on the bottom, or the cells could only be exchanging with their neighbors, so it could be only cell to cell. And so we have the possibility of secreting or absorbing only on contact with particular other cell types. The other thing that we have to do with secretion is we have to determine, just the way we did with our boundary conditions, and this is something we didn't play with, we could have played with Connor's example. We used fixed value boundary conditions. And I had you in some of the homework problems play with fixed flux boundary conditions, constant rate rather than constant value. And when you work with constant rate of sources or sinks, life is a little bit more complicated than with constant value. If I imagine a hose pumping water into a tank, and the tank doesn't leak, the equilibrium amount of water in the tank is infinite. The water just keeps going up. If the tank has a leak, and I pump water in, and the outflow is proportional to the amount of water, then the level in the tank will go up until the flow in balances the flow out. And the equilibrium value will be the inflow rate divided by the outflow rate. That's actually more or less the same analysis, dimensional analysis we did before. And so if you have constant input rate rather than value, the concentration could go way above the amount you're putting in or below, depending on the rate at which you lose the chemical. And that means that constant flux methods tend to be relatively sensitive to the parameter values in the situation, so they're relatively unstable. On the other hand, there certainly are times when that's what's happening. If I have a pump that's pumping glucose into my dish, I really do have a constant influx of glucose. So I want to model that. If I have a cell that is secreting something into its environment, and the secretion rate doesn't care about how much is already in the environment, then I want to model that. And so I do have to think carefully about whether I have constant value or constant flux for secretion and absorption. So if I use the secretion plugin, I have to give the name secretion plugin. I need to say what the field name is from PD Solver. I have to say, in this case, who's secreting, what kinds of cells are secreting, whether that secretion is everywhere or only when I'm in contact with other particular cell types, and then whether this secretion is of type constant concentration, constant rate, and there's some other options. If I'm going to specify things in Python, I just need to say that I have secretion, and I don't have to do more than that. If you want to do absorption, again, you'll have a bunch of options. And as we found out the other day, not all of those options are in the Twidit menus, but a representative selection are, and you can hand edit the commands to make that work. So if we're going to say have cells absorb oxygen from the oxygen field, we'll go to the Twidit steppable, use that Python pull-down menu, and then there's a secretion uptake track amount. And again, inside cell at boundary, track amount, inside cell at boundary with contact, track amount, inside cell at center of mass, track amount, inside cell, outside cell at boundary on contact. There are a lot of them. As I say, not all of them will show up in that list. I think in the newer version of PyCompuCell that you have downloaded for desktop, it should all be there. But I know in the Twidit for on NanoHub, there's some that are missing. Okay. So again, coming back to constant rates of secretion, let's ask the question, what is the concentration of the chemical, assuming the boundaries aren't doing anything? So this would be periodic boundary conditions. If I have this diffusion equation, and I've already jumped ahead a little bit because this should be d by dt of the concentration equals the diffusion constant, second derivative of concentration, minus decay plus source. I've set the time derivative to 0 already. I'm now going to set the spatial derivative to 0. And what I'm left with is 0 equals minus d of c plus u, which means c is equal to u divided by d. And so if I have a constant source rate, flux of chemical, and a decay rate d, and I realize I use gamma some other places instead of d, the equilibrium concentration will be u over d. So why don't we do that? Let us try this out. Let us create a simulation in which we have 100 by 100 lattice. We'll have a chemical field called oxygen. And we'll say the thing that's doing the secreting is medium. And we'll say it has a secretion rate of 0.2. Diffusion coefficient is 1. And the decay constant is 10 to the minus 3. So why don't people give that a shot and try that out? And you notice I have carefully not said whether you should have other kinds of cells in this or not. I'd like people to try that. And I'm not sure I should say in this case, is it clear what I'm asking you to do? I'd like people to interpret this like a piece of postmodernist music where you have to guess what the score means. Something by Xenakis, maybe. Or Crumb. They give you a piece of paper with a squiggle and they say to the orchestra, play the squiggle. So why don't people give that a shot? And if you think about it for a few minutes, and if you're desperate, we'll work on it together. Okay? And if you're completely lost and have no idea where to start, that's okay. Then come. You can text me or text Juliana. If I remember properly, I think I have a hint in the next slide, so I may jump to the next slide just to check. And if it's there, that's good. If it's not, we'll come back to this slide. No. So that was the answer. So all you have to do is repeat what Connor did, which is create a simulation, load the diffusion solver, and then hand edit the secretion rate of medium and the diffusion coefficient to be as advertised. Since we're going to run out of time, I'll show you my solution here to begin with, but you can come up with other solutions. Something to think about is that when I gave you the equilibrium concentration, you'll notice there was no diffusion constant in it. The equilibrium concentration depends only on the amount coming in, the amount going out. It doesn't depend on the diffusion constant. Okay, this is more or less what you should get if it's working. I notice here that this should say periodic, and the periodic boundary conditions are actually commented here, so they have to be uncommented. And since there are no cells, medium is everywhere, so it's sort of a boring simulation. Did that work for you, Connor?
I did that for the homework. Doing it the second time now, I realized I forgot to initialize one of the steps, so I had to start over. So I'm one step ahead in my previous code and one step behind now. I will say I tried to do what you mentioned at the start of class, where I said that when I tried putting cells in it, it was crashing. It does run now, but I get a really, really weird result. So I think I made a bigger mistake elsewhere. But this step that you just went over, I was able to get.
Great. Anybody else? Joel, how's it going?
Yeah, I have a weird result. I don't know what I'm doing wrong. But my medium stays the same, and I couldn't see your simulation. But it's like getting different rates. Right now I'm in 100 or something like that, but everywhere.
Okay, what about other people? Josh? We lost you.
I think mine's working correctly.
Okay, great. Do you want to show us?
I'm really sorry, but I actually have to go.
Okay. Yeah, we're over time. We started a little bit late, so I apologize. Does anybody want to end the class by showing us a result?
I think mine is wrong, but I can show.
Okay, let's take a look at
It's
it. and
just
then
wrong. Hold on, let
we
me
can
share.
help you debug.
Okay.
And next week I'll try to start on time so we don't run up.
Yeah, you see how the just this is changing. I don't know what I did.
Wait,
We
is
should.
it changing when you're moving your mouse?
No.
Okay. That seemed like that. Okay.
It's
So
just,
you
I
have
think
a
it's just jumping.
spatially. Sorry, go ahead, Connor. Somebody?
Oh no, I was just responding to Julian's comment. I was gonna say it looks like it's jumping discretely, which is why it's looks like it's the mouse.
I think it jumps discretely because it's saying at every 10 Monte Carlo steps. That's why it's discrete. But
So-
I think I'm putting like secrete, the medium to secrete, and it is secreting. You can see that is going up, right?
Yeah.
But I don't know what I did.
Well, what is the secretion rate and what's the decay rate?
Yeah, okay. That I think it's my mistake. Hold on, let me stop
No,
sharing.
I don't think you have a mistake.
Okay, let me
This
stop
looks
sharing.
perfect. Well, no, it's okay. Look at you don't need to go, you can leave it, you can go in player. It's all right. Open the diffusion solver on the right, on the left, sorry. There. And look at the diffusion data and the secretion data. Okay, good. So what is the global decay constant?
1.
Decay constant.
Oh, yeah, 0.001. Should
Fine.
I
And
make
what
it
is the
bigger?
secretion? No, you don't assume you're doing something wrong until you figure it out. You can calculate what you get. So what is the secretion rate?
0.2.
Okay, what is the equilibrium concentration that we just derived?
Just a second. 0.2.
Divided by?
Divided by 0.01, right? Because it's times 1.
0.001, which is?
Math, help me.
What is
Calculator.
0.2 divided by 1,000? You don't need a calculator to do that.
Help me. I do. I do.
We can blame this on the fact that it's dinner time, so low blood sugar.
Oh, my head. 200. Yeah. So it
Okay,
was
good.
okay. So it
So
should
now
stabilize
run the
at 200.
simulation and see what you get.
Okay. I had a big day today. My brain is not functioning.
All right, let's see what happens. If it goes to 1,000, I'm wrong.
Oh, I see. You're saying the reason it's jumping still is because it's approaching.
It should stabilize at 200.
Yeah.
Stabilize
right.
at 200.
I
Yeah.
jumped a lot of
I
those
won't
times.
promise that it will. And
This
the
is
time
very
and the
suspenseful.
time it should take to do it is 1,000 time steps.
I don't even know where it is. Hold on a second. Let me
Oh,
No,
bring
no. We can. It's on the axis. Yeah.
5,000. Oh yeah, it's on the axis.
All right.
Yeah, it's stabilizing. It's getting slower.
Well, it looks like it's still going up. So you may make a liar out of me yet.
Yeah, but if we look at the min and max on
Min
the
and max is
right,
almost getting to the yeah.
it's not even 200 yet. But
Yeah.
yeah, it's ever so more
Mhm.
slowly getting.
Mm-hmm. Yeah. It's almost like we're getting to the
Yep.
lower part of the
It
function.
may overshoot it. It may overshoot it a little bit. As I say, the numerics aren't always
Yeah.
perfect though.
Now it's stable. It's stable. It's not changing, and the steps is still going.
No. Okay. So okay, good. So now you can go into that editor. Let's make the secretion rate half of what it is. It should go down to 100. You add 0.2, 0.1. See what happens.
Oh, I have to press enter, I guess.
Yep.
Yep.
All right. Now it's going down.
It's coming back. Cool. I thought I
So
was wrong.
So it works.
Joel, if it makes you feel any better, I can show you something that's very clearly wrong to make you feel better about your code that seemed wrong but was not.
But this is a reason to do a little bit of simple algebra, like
Yeah.
calculating what it should be. And I understand that it seems easier to write computer code than to divide things and multiply. But it's good to develop at least a little bit of intuition about these things first.
I question myself even on the correct answers. That's the problem, I suppose.
Well, I think it's fine to question yourself. It's good to be skeptical. But it's even better to be skeptical when you have the ability to do the computation.
Yeah, and confirm, right? or deny.
Exactly. Okay. Okay, good. Well, thank you both, Connor and Joel. You were very, very kind to be the guinea pigs today. Drew and Jackson, next time you may be on the spot a little bit, okay? I